It's good to start another great tech talk. And in her absence, we want to thank Helene for arranging this. Just a bit on LaRSSA while we're waiting for the other people to join, give them a bit of time still. LaRSSA has 2 tech tours that's fully booked at this stage. So a lot of us are going to have FOMO, especially on the Western Cape tour. And then the one to Voorspoed Mine as well. Other things that LaRSSA are going to be involved in is the National Department of Agriculture, ARC and SANBI talk on rangelands. So stay tuned and watch our email correspondence, or if you are in the WhatsApp group, just look out for the registrations for that, because there's a great drive towards rangelands and the natural capital entailed in good regenerative rangeland management. Then LaRSSA is going to be at the Regenerative Agriculture Conference in Reitz, 3 and 4 March. So if you are seeing Teresa there with a LaRSSA shirt, go and say hi. Make sure you join the LaRSSA community in some or other way. We would like you to be a member, but that's all up to you. And then LaRSSA will also be at the Coltech Colloquium on the 28th of May. That's a registration, free attendance, and the link will be published later if you want to be part of the discussion regarding coal and better, greener technologies. I think I'm going to wait for 2 more minutes and then we'll start the talk. Just, while we're waiting, a point of order also, please write your questions and answers in the Q&A part of the Teams tabs at the top, and then in the chat you can state your name and from where you are, and just telling us a bit more of your interest in this biotechnology talk. And we are excited to hear about everybody on this talk as well. So LaRSSA is very excited to have Blueplanet South Africa to give us a talk today, because what if the solution to our biggest environmental challenges wasn't more chemicals, but better biology? Blueplanet is South Africa's leader in eco-friendly biotechnology, replacing the old world chemical industrial cleaning methods and waste management with high-tech microbial solutions. So today we are privileged to look at how they are cleaning up wastewater, water, waste, all kinds of industries inside out. And I want to welcome Rowan McDonald, the specialist in bioremediation and nanobubble technology from Blueplanet, who will take us through most of the slides, as well as Heiner Dominic, who is the founder and managing director of Blueplanet. And I think this is where I shut up and give Rowan the mic. Thank you, Rowan.
Thank you, Anya, and thank you everyone from the LaRSSA team for this opportunity. As introduced, Rowan McDonald, Head of Technology for Blueplanet and Bionomics Group. So my job is to basically handle the designs and speciality in the use of some of our core technologies. As a company, we're a specialist bioremediation group. Our experience lies in blending biology and technology to restore water, land, and environmental resilience. I'm going to take you through sort of some of the South African story, the status quo as we see it, and lead into why we developed something called the Bionomics approach. I'm then going to do my best effort in a short space of time to give you sort of a 101 class on what is nanobubble technology and microbial remediation. We'll then run through some case studies and applications. We'll keep that local and briefly cover some challenges and cautions. So what we're about is really bio-based restorative interventions to man-made impacts on the environment. So we do this by essentially intensifying water management and food systems. A lot of what we do is focused on water, but we also work on soil. And what we do is really accelerate the water cycle and focus on the biology, how we can support it and how we can optimize it to cycle nutrients and degrade pollutants. So we do this, it's all about acceleration, it's about scalability and keeping things biological. So a couple of facts. I think these are fairly well documented. Some of them might not be new. Non-revenue water is at about 2 billion cubes a year. Interestingly, our invasive alien plant issue consumes and wastes a similar amount of water each year, and that is projected to scale sort of 2x by 2032. In terms of our river systems, I think it's well understood the crisis we're dealing with there. 65% and growing of our river systems are in ecological collapse or highly threatened. This one I didn't have on my radar until I looked this up, but South Africa has lost 35 to 50% of its wetlands. So if we think in terms of our natural sort of water purification and flood buffering, we've lost half of what we had over time. Another well-known fact that's sort of very integrated with a lot of these issues is 64% of the water treatment works are listed critical. That's back from 2023 Green Drop reports. We eagerly await the new ones. It's anyone's guess if we're faring better or worse there. In our line of work, we only seem to see the bad ones, and pretty much doom and gloom in terms of the wastewater treatment infrastructure. So looking at those figures, who actually was compliant, only about 13% of municipal wastewater treatment was meeting compliance. Microplastics, another big complex issue. We have a lot of sort of primary issues to deal with in terms of wastewater and the environment. Microplastics are another demanding one. Sad to find that in a recent Vaal River study, 100% of soil and water, sediment and water samples, microplastics were identified. So the way we see things, particularly in Gauteng, we have a very highly integrated network. Obviously, water is a cycle all over the globe. But if we really just put a lens on the sort of Gauteng region and how tightly integrated our wastewater to surface water to drinking water operations are, you start getting a picture of how serious bad wastewater is or non-compliant discharge. So the stressed wastewater infrastructure is having a daily impact on environment. This in turn affects ground and surface water in terms of sediment accumulation, eutrophication, toxins, and pathogens. We are familiar with many drinking water plants that are simply not able to cope with the biomass in the eutrophic water bodies that they draw from. We're seeing drinking water plants cease operation and shut down because they are not built to deal with the loading that's coming in. We're working on addressing these, but it's a case of there's very short-term issues in terms of the ability for drinking water plants to actually treat the water that they're abstracting, but so to have the sort of longer outlook on how and why those surface water bodies got to that point. So, restoration, rehabilitation, optimization is needed both on the environmental surface water side, but so to the treatment process that has to deal with those water quality issues. Sadly, these large infrastructure projects, particularly wastewater treatment plants, work on a very large, long time scale, from budgeting, tendering, and procurement and construction. You're typically looking at 4 to 10 years. Most of this wastewater infrastructure has got sort of 10-year timelines behind it. We can't wait that long for a wastewater treatment plant to be upgraded and compliant, whilst daily impact and stress is occurring on the environment and our water use systems. It's estimated that about 900 billion is needed by 2030 to sort this infrastructure out. 2030 also happens to be the time where demand is going to exceed supply. That's been around for about the past 6 years. So what's needed? Obviously, compliance and enforcement. There's some fairly healthy laws and regulation out there. It would be nice to see stronger compliance and enforcement. Water treatment plants, particularly wastewater treatment plants, we're looking at how do we intensify, how do you make the best of what you have without the long wait for those civil upgrades and massive sort of 10-year construction plans. Is there something we can do in the interim while you wait for the major infrastructure sort of civil works to implement? We're needing solutions in the environmental and surface water side. The eutrophication and pollution of these lakes and dams is critical because of the scale of these systems. It's a challenge, but the cost of sitting back and watching it happen is even greater. So we need novel, rapid deployment, preferably low OpEx solutions to drive change in this water management cycle. I believe another good approach is supporting decentralization efforts, both in terms of municipal water infrastructure, perhaps own build operate systems for the private sector, as well as a big drive to sewerless sanitation systems. South Africa is quite renowned for its innovation. We do have some great minds in this country in terms of engineering and science, and it would be nice to see more support in the R&D and then a sort of unimpeded path towards deployment. R&D is important. Establishing true success and getting validation behind various treatment approaches is important, without having too many stumbling blocks to actually getting these solutions in place and making a difference in the environment. So briefly on us, I don't want to make this a long story about sort of our company, but there are some interesting points as to what we had to deal with and sort of how we got to the point where we're working on some of the largest remediation projects in the continent. So about 15 years ago, Dominic, who's on the call with us, our CEO, had come back from Europe back into South Africa and had established some businesses and designs around packaged plants, wastewater systems for remote areas that could recycle water. Those packaged plants, very novel, great idea, worked really well, but occasionally you have severe challenges in terms of what the user base has thrown down the drain. And, you know, this is an issue that municipalities and cities deal with on a larger scale. Debris, foreign objects, etc. are the obvious ones, but what's a little bit more hidden is the cocktail of chemistry that we all throw down the drains. Soaps, drug residues, pharmaceuticals, etc. So fats, oils, and grease, surfactants, and even these hydraulic stresses from high consumption periods. So when you're in that space, you tend to have what we call bug salesmen come and provide you some magic biology in a bottle or a bag, say throw this into your plant or throw this into your lake, and the hohos are going to eat up the problems. So that was tried, many different products, and eventually discovery was made in terms of quite an advanced formulation that really did make change. It stabilized the plant, it stabilized the biology in the process, despite all of these challenges. The use of that product then grew outside of the water treatment space and started getting into soil applications, larger water body applications, sewage spills, etc. The success was quite incredible. And we put that down to the product not just dealing with degradation of carbon compounds, but also dealing with the nutrient cycle, the ammonia, nitrogen, phosphorus, salt, and sulfur species. So the work got bigger and bigger. Ponds became lakes, packaged plants became municipal works, and just before that, Blueplanet South Africa was born back in 2015, and there the work got more complex. We're working strictly with biology. Biology works to its own time frame. It is not rapid. It's not chemical where you throw in a chemical, a reaction occurs and you get a result. Biology is a process and a cycle. So a client is asking you to tackle a larger and larger, more complex issue in the environment where you don't have much control, and they didn't want to wait, let's say, 18 months for thousands of cubes of sludge to be digested. They wanted it happening in a shorter time frame. Because it's biology, you're beholden to temperatures, salinity, pH, etc. So the more you can steer those conditions to where you get optimal microbial performance, the better off you are. And that led us to how do we get more oxygen into these systems? It's either in a wastewater treatment process, and it's a lot more complex in a large water body or river. So aeration, as is commonly known, could offer some support, but it really wasn't delivering what we needed, particularly in challenging conditions or larger projects. I discovered something called nanobubble technology back then. There was an early invention of this emerging tech. We put it to work in some projects in wastewater and environmental surface waters, and it gave us a lot more than we were expecting. We like to call it the 1 + 1 = 11. It was more than the sum of its parts. The combination of the microbes we were working with and the nanobubble technology really accelerated the outcomes for rehabilitation. That early invention had some technical challenges in terms of the reliability and the engineering, obviously sort of being a draft one of a novel tech. By that point, we were quite hooked on what it could do for us and our clients and overall environmental remediation. So the obvious idea was to go and search for the best and the most scalable solution out there. And cut a long story short, we were selected by Moliere USA to represent them in South Africa. It's the leading nanobubble technology application and science company in the world. And we've been working with their tech and integrating it into our approach over the past few years. So this led to larger and larger projects that were perhaps not feasible, became feasible, timelines accelerated, OpEx budgets reduced, and the continued development of how we integrate these various technologies got to a point where we were able to start packaging them and still to this day further refine and advance on that theme. So those learnings and intellectual property, which has been developed by Heiner and team in South Africa over the past decade, is known now as the bionomics approach. The ethos of which is less chemistry, more biology. The vast majority of issues we're tackling come down to biology and how we're able to steer that in the right direction, even in challenging environments. Most of these applications can become more functional if they're aerobic and provided oxygen. We need to be smart with how we do that and efficient. These tracks are versatile and prescriptive, so no project is the same. But the principles and the core technology set are there, and it's how we assess, manage, and steer the outcomes of this is where that intellectual property is. It's obviously very data driven from inception and study of a particular project or task right through to project monitoring, which will adjust the prescription, and finally the outcomes and rehabilitation reports thereof. So we've stacked that into a few key tracks. Bionomic balance is for lakes and dams. This is where we recalibrate the trophic status of an impacted water body. So something that's become eutrophied or overwhelmed with pollution may have stopped its nutrient cycling. It may have algae blooms, accumulated sediments. So we work from sediment to surface, recalibrating and restoring natural function, basically reversing the aging of that system and getting its core cycling to occur again. We do similar things in rivers and streams called bionomic flow. This is normally about dealing with direct pollution impact, odor, and toxicity. So we get that river to breathe again and be able to reduce its pollutant load that it's receiving. Bionomic dynexis is focused on municipal and industrial wastewater treatment process. That's where we get that plant to essentially do more with less. We optimize aeration and biology. We reduce OpEx and or bring that plant into compliance without drastic mechanical, electromechanical, or civil changes. So we do this in lagoon systems, we do it in activated sludge plants and membrane plants. Bionomic remedia is pollution cleanup in soils and water, so in situ remediation from chemical and hydrocarbon spills. Bionomic Thrive is these core sets of technology applied into agriculture and soil health. So the core technology stack, which is then sort of custom integrated and prescribed to an application behind this, is our microbial technology, which is known species. We know their function, we know their habitat, and how to steer them into an application for soil, wastewater, agriculture, or similar. Nanobubble technology, which we'll get into, is the accelerator to all of this, where we can do gas diffusion and create these nanobubbles which stimulate the microbiology. Bioengineering, we bring this particularly into river and lake solutions and pollution control, anything from biologically active weirs, floating wetlands, in situ bioreactors, creating massive amounts of surface area in systems so that we can have beneficial microbial habitat to restore biodiversity. In terms of the data around these projects and reporting, we work with everything from sonar scanning lake beds and sediment and doing hardness and vegetative mapping through to real-time in situ monitoring in pipe, in plant, or in lakes and rivers, including sediments, drone imagery and surveys, laboratory analysis. We bring this all into a dashboard for assessment, informing the direction of the project, and ultimately the final reporting. Data is certainly key. So the kind of work that has been achieved with biology and nanobubble oxygenation is anything from large municipal that have suffered decay and degradation or not meeting compliance, river systems that have received pollution for a long time and need rehabilitation, large-scale lake eutrophication management, agriculture, and more. We'll dive more into some specific applications later. Okay, time for a little bit of schooling. These are very complex topics, and I'm sure I have a very mixed audience of skill sets here, where some of this might be well understood and some less so. I'm going to do my best to keep it short, but I'm going to raise a few pertinent points around what to expect from microbial remediation, what to look out for. So just briefly, what we believe is important and why we've had so much success in the market with our particular product set is we are working with pure cultivations of organic bacteria. We know what they are. This is not some concoction or cocktail that's been created years or decades ago that keeps on fermenting. These are individually identified, sequenced, and libraried microbes. So the industry, microbiology, there are major reference libraries around the world, sort of species or genome banks, where microbes are studied and identified, what environments do they exist and where in the world. We work with select strains that are well understood globally and exist naturally in water and soil systems everywhere. We've refined this. Our Professor Dr. John Wong has refined this over the past 25 years. We've selected the most functional species and speciation that is acceptable around the world as a naturally occurring bacteria. The way we then produce and stabilize those is our sort of secret sauce. It's quite difficult to get different bacterial groups to coexist, ready for you to use into an application. So we've got a highly scalable production process, as well as stabilization. So we know what's in there, we know the counts of what's in there, and we have batch identification and validation around what we've produced. Importantly, there's a lot of bacteria products out there that are sort of these mixed inoculums of you're not sure what. They batch ferment them, they feed them molasses, and you get a lot of growth, and by all accounts, maybe quite a high plate count from bacteria. The limitation to that is you're kind of training your bacteria to go and eat molasses, which is maybe not what your field work is about. We use specific column recognition compounds. So the bacteria are given specific food sources to support their growth and habitat. We then grow these out, blend them together, and importantly, stabilize them. So we have a microbial consortium with a 2-year shelf life, which eases logistics and storing, and essentially ready for field use without the fear that you have an unknown in your product or an unknown quantity and quality. So why do the so-called bugs matter? In terms of water treatment and the water cycle, chemicals and mechanics alone are not everything. In a wastewater treatment process, they're important, but it's really the microbes that are doing the work for us and converting complex waste, mineralizing them out to things like CO2, nitrogen gas, and water. So it's really the biology that is responsible for most COD, solids, ammonia, sulfide, and phosphorus removal. And we can talk about this universally from wastewater process through to lakes and dams that receive this loading or have natural loading from animals, flora and fauna. Bacteria are basically fairly sensitive to the kind of environment they're in. They have core functions, and this will be dictated by oxygen levels or lack of oxygen, type of food source. They can be quite competitive with each other. Temperature is another variable, as is pH and even ionic chemistry. So with all these considerations and sort of all these options out there with microbes or enzymes that could be used, what I encourage us to think about is what is that product doing in terms of nutrients and the breakdown of the kind of pollutants you're looking at. The vast majority of these products work purely on the carbon cycle. All right, these are your organic degraders. These are the easy microbes to work with. This is what's fairly abundant. The question should be around, what about the nitrogen cycle? Breaking down organic sediments and sludge will typically release ammonia. How do you deal with that? What about the sulfur species that come out, as well as phosphorus? So talking about these environments, we have temperature classifications. Most of what we're going to be working with in our fields in soil and water is mesophilic. And we further break that down into, does it work off of photosynthetic energy? Does it require a lot of oxygen, a little bit of oxygen, the facultative species, or is it anaerobic? There's sort of no class leader here. It's good to support all of these microbes. Aerobic microbes do certain work for us faster. Anaerobic microbes are equally as useful in terms of reducing something like nitrate. So what's its food source then? This is in terms of what you might need to introduce, or at very least understand your target. Quite often you're trying to look after all of these. So ammonia, phosphate, the various nitrogen species, the organic carbon, and the different types of carbon. So I'm going to get into enzymes now, because quite often microbial products, or what people might refer to as bugs, bioenzymes, there's a lot of confusion here. So bacteria are living cells that can consume or cycle waste. All right, they replicate and grow if the conditions are correct and if the food source is available. Right, an enzyme does not reproduce. An enzyme is actually the production of a bacteria when there is not soluble food available. So if you think about the sediments in a lake or sludge in a lagoon system, you have sort of coarse food, that's the sludge and particulate matter that you can see. A bacteria cannot actually make that disappear or eat that. A bacteria doesn't really have a mouth. What the correct bacteria in the correct environmental conditions will do is produce an enzyme that does that first breakdown and solubilizes that food source for the bacteria to then take over. So if you only apply an enzyme, you will start solubilizing and releasing carbon and nutrient. But what happens next if you do not have supporting bacteria to then complete that process? So just a word of caution, yeah, be careful with enzyme-only solutions, because you're not completing the cycle. In fact, sometimes you can have quite severe spikes of nutrients, which create further issues. If we're to compare this with chemicals, chemicals, is a reagent-driven process or reaction. It could be oxidation. We could use them for coagulants, pH adjusters. Chemicals can be complementary to a biological process or inhibitory. What is fairly consistent, though, is chemicals don't replicate like bacteria. They don't really harvest energy from the localized system, and you are constantly needing to introduce those chemicals to drive that reaction that you need. So it's not to say all chemicals are bad, but pick your target. What are we trying to achieve? If you're trying to solubilize and reduce suspended solids, sludge, and take care of the resulting nutrients, biology is going to be your friend if you understand the way to apply it and how to apply it, or at least support it in a natural environment. So talking about the sort of workforce of these little critters that degrade waste and pollutants, we get many different types, as I showed you. This is the speciation we work with in these various projects I'm gonna talk about. So we have Rhodocynonarmus plastris, which works in aerobic and anaerobic environments. It actually uses photosynthetic energy as well. Very, very useful for a wide range of organic compounds like phenols and hydrocarbons, and can also sequester phosphorus. We then have the Bacillus army, which does a lot of our organic degradation and carbon-based work and produces some very, very useful exoenzymes. Licheniformis is a denitrifier to complete that ammonia-nitrogen cycle. And very importantly, the nitrifying species. If we only had carbon degraders, if I can put it that way, you would stand to have initial degradation, release a lot of ammonia nitrogen into the system, and then what else? So some of these products could create ammonia nitrogen issues. Unfortunately, we've got the speciation to then deal with that. Nitrifiers are notoriously slow growers. So if we look at the sort of workforce species of Bacillus, if you had one bacterium in the right environment, 5 hours later, you'd have 1,024, and so on. The nitrifiers, which are more sensitive and slow growing, one nitrifier would become 32 in 5 hours. So it's important to have those available, given the conditions they need to work to complete that 360-degree cycle of how pollution, wastewater, and even soil health works. So how do you put this to work in the field? Ready-to-use liquid products. This is an easy platform. You get to apply liquid to wastewater process, irrigation systems, surface water systems. And what we've learned over time is obviously the larger the project becomes, the more and more biological support we need. So quite uniquely, we offered the Southern Africa market the ability to batch prepare on site. So this is how we scale up this biology. So we build a bioreactor on site and essentially use the liquid products and some powder products as an ingredient to then grow and multiply the use, the amount of microbes that can be applied to a project. So we're not transporting water around the world or around the country. We're able to do that rapid growth on site, and even acclimatize microbes to specific conditions. So if we're dealing with pH extremes, salts, hydrocarbons, or other toxins, we actually train this microbial community to tolerate and work in that environment. So we customize it to the field application. So I'm sure a lot of us have had some sort of exposure to purchasing bugs or bioenzymes or some kind of bacterial product. You may have had success, you may have had failures. Unfortunately, there are quite a few failures with these products in the market. What I think is misunderstood or not regulated in the marketplace is an understanding of what is in that bottle or that pill or that tablet, and how to get it to work. So my advice is, if you're looking at something to use in a particular application, get an understanding of what the species are. Any service provider worth his salt should be able to give you a list and an analysis of what those species are. Hopefully they explain to you what each of those species are for. If not, you could sort of do your own research on what the functions are. Are they standardized? Are they part of an internally recognized genome library? Have they been sequenced? Or are they some sort of GMO or unknown? We would apply caution if there's some unknowns there, or some unexplained species, or something foreign to your particular country or application. So the simplest questions are GenBank numbers and biosafety levels. What is in that bottle, or what is in that powder or tablet? Other questions might be around the pathways. So most of these products are sort of heavily carbon focused. What happens with the degradation and breakdown of your pollutants and carbon? How are the rest of the nutrients dealt with? What happens with sulfur species? What happens with ammonia and nitrogen? Do you have the rest of the sequence to deal with the outcomes of what happens with that? And then how viable is the product in terms of storage and field use? So if it was not stabilized at production, that bacteria would assume to be live and then in a constant state of death and decay since the date of production, or is there some kind of stabilization? Most of your powdered products are going to be stable. It's the liquid ones that you should question in terms of viability. All right. Sorry if I try to condense too much into that one, but yeah, I'm trying to cover a lot of material here. So that's a sort of focused look at the world of bioremediation, bioaugmentation technology. There's obviously a lot more complexity to it, but I think what we've shown in field work in South Africa, work with universities and municipal departments, is it can work if you understand your expectations, your environment, and your species choice. Getting those things right is critical. Managing the expectation and supporting the biology. And it's not just about throwing lots of unknown biology at a problem. There is some finesse to it. All right, so nanobubble technology, this is an exciting one. This is my field. I get very, very passionate about it. This is what stepped up our game and our scale in terms of remediation. We now, the Bionomics model and the use of nanobubble technology has now moved on to reestablish Blueplanet Germany 3 years ago. We're tackling challenges in Germany, New Zealand, other parts of the continent too. So the sort of integrated approach of how we work with biology and support it with nanobubble technology has really grown leaps and bounds. what got me into this field, besides this sort of infectious passion from our founder Heiner, is this graph. When I got into it, you obviously do a knee-deep dive on publications. What are the scientists saying? What are the institutions saying? It's a fairly new class of science. 20 years ago, there were still arguments about whether nanobubbles really existed. They were discovered in nature first. They bent a few of the gas rules at the time. They shouldn't exist. They shouldn't be stable. So it was the age of discovery, and then the age of engineering and application. That's where we are now. So industry started learning, how do we make nanobubbles? How do we get them to work in environmental water treatment process, and even medicine? There's a lot happening in that space. So this graph's old here, but you can see the ramp up in terms of publications. There is something new coming out all the time now. A lot of the foundational science is established, and we're more and more learning about niche applications in water and soil. The graph on the right is actually just an extrapolation I did because I haven't got an updated graph here. But as you can see, the science and research is really compounding. I'm gonna do a quick lesson for those who are not familiar with oxygen transfer or aeration. So if you do want an aerobic process, or you're adding oxygen to an application is going to assist that biology, there's a few different ways to do that. The most common of which is sort of large mechanical aerators that like sit at your wastewater treatment plant. Huge amounts of energy, not very efficient at getting aero oxygen into water. What is then the next advancement on that is some kind of diffuser. If you can picture your aquarium tank, you've got some sort of bubbling air stone, and those bubbles rise up through the water column. And the amount of time those bubbles are through that water column, that's your chance to try and diffuse that gas into the water. So deeper water helps because the longer the bubbles are there, but you're using a lot of energy and you're always having bubbles pop out. So diffusers are generally only getting about 4 or 5% of the gas into water per meter. And there's a lot of wastage in that. Cones or species cones, this is something the aquaculture industry uses. Very complicated to design, extremely power hungry, and not something you're going to see in a wastewater treatment plant or environmental. Sparges and venturis, these are better. Some of the most well-designed venturi systems I've seen are hitting sort of 20 or 25%. The rest of that aero oxygen is wasted back to atmosphere again. Nanobubble technology gives us the ability to dissolve over 85% of that gas into liquid. We're doing this with low energy, simple maintenance, and across a wide range of applications, highly scalable. So efficiency is one of the reasons we use this technology. To scale this, we're familiar with macro and micro bubbles. All right, micro bubbles are going to be in your soda, in your Coke, and they will eventually pop out of that liquid. Ultrafine bubbles create a sort of cloudy water. These are much smaller bubbles. They hang in that water for a little bit longer, so they have a better chance of trying to transfer their gas content into the liquid. Nanobubbles, on the other hand, are invisible to the naked eye. So this picture is not to scale. We are talking about a gas bubble a similar size to a virus or bacteria, right? We could line up 1,000 nanobubbles across a strand of hair. We will fit trillions and trillions of these nanobubbles into half a glass of water, right? So it's difficult to imagine these nanobubbles, particularly when we can't see them. but just realize, we could fit hundreds of thousands of these bubbles inside a conventional bubble. So why is that interesting? One of the most defining behaviors around nanobubbles is the fact that they are neutrally buoyant. They don't actually rise out of water, right? So that increases our ability to have gas retention and very efficiently get oxygen or air or some other gas into that liquid. They're highly stable, okay? They don't rise. They have a hardy surface, high internal pressure. They are electrochemically active, negatively charged, and hydrophobic. So I'm throwing a lot of terms around here, but really what it means is the bubbles repel and push apart. They will not join up and become a larger bubble. They mobilize really well throughout the water system, the tank, the lake, or the application. They're happy to be at depth in a system. So if we look at lakes or lagoons where you have sludge and sediments at the bottom, that is typically an anoxic process down there. It's slow, it produces smelly gases. By introducing nanobubbles, we can turn those environments into microaerobic environments, accelerate the decay of that organic matter, switch off the modes that produce those noxious odors. So we're able to do things with this platform in terms of functional nanobubble behavior and oxygenation that we simply couldn't do 15, 20 years ago. So it is driving various benefits in water treatment management and environmental that affect both the physics and the mechanics of treatment, the chemistry and the biology. So how do we make them? With a Mollier nanobubble generator, there's really three core components to this. We need to circulate the liquid. We need to bring the liquid to the system. So we will have a water pump abstracting from a lake or a tank, or be part of a water treatment process that is pumped. The nanobubble generator core does not actually have any moving parts or wear and tear. To that core, we connect a gas source. This could be air, it could be oxygen produced on site, it could be tank oxygen, ozone, hydrogen, nitrogen, CO2, whatever that gas is that we need in that process. We introduce it there, and we get this dissolving of gas and production of nanobubbles. So the size and scale of this will vary based on application. But in terms of efficiencies on the energy side and mechanical side, it's quite incredible. So that technology is really doing two things in water management. It is dissolving your gas of choice hyper efficiently, 85% plus transfer. That is quite often a reason to introduce this technology on its own, is for its ability to provide that gas very, very efficiently. Secondly, and possibly more importantly, is the production of these nanobubbles, which behave more like active particles. We're able to enhance and manipulate separation processes, accelerate biological function, strip out contamination, sequester inhibitory compounds. There's a laundry list of what these bubbles are doing beyond just their gas provision. Their physical traits, behavior, and electrochemical reactions are immense. So we've got to remember this is not just about oxygen or not just about a bubble. It is about the way these bubbles behave and how they're able to enhance various processes. The other thing that helps nanobubbles be so functional in biological and separation systems is obviously the scale, which results in this incredible interfacial surface area. What I mean by that is the interaction potential, the ability to react with a fine contaminant, a solid, a nutrient, a bacterium, a sediment. There's so many of them, and they are actively charged. They want to interact with the components or the stuff or pollutants we have in the water and or soil. So if you had a normal gas bubble, 5 mil diameter, you had 1,000 of those. If we calculated the surface area of conventional bubbles, you'd be at 0.07 square meters. If we took the same amount of gas that was in those 1,000 5 mil bubbles and converted them to nanobubbles at 100 nanometer scale, we would have a surface area of almost 4,000 square meters. So this speaks to the efficiency around the gas, but so too then the physical, chemical and biological interaction potential you have in a particular application. So the scale completely changes here. We're doing essentially more with less in terms of that gas and mechanical input. So the world of nanobubbles is very exciting. I would encourage you to look up a few papers, particularly in your specific fields. Please do reach out to Hannah or myself after this if you've got questions for your field. And chances are there's probably a paper on it somewhere by now. It's used, like I said, even up through to the medical industry and drug delivery, which is more emerging. But we're seeing very interesting applications like surface cleaning, where the hardy nature of that bubble itself is actually able to scour and clean a surface like a tank or pipe or food material without chemistry. Just using an air or gaseous bubble is actually giving us the cleaning effects. Water mobility, dispersions, a lot happening. We tend to focus on its use for gas transfer, microbial process and stimulation, and what we can do in separations. Because really, these are the real drivers behind water and soil rehabilitation. Right. I'd like to talk a little bit about the water cycle and surface water. So we've got a lot of impaired water bodies, not just in South Africa, but even Europe and US are also facing this crisis. Man-made activities have sort of overwhelmed the natural cycle in these water bodies. We're seeing it happen in real time in the country, things getting worse, but it's also been happening for decades. There's two key considerations when we talk about an impaired water body. We can see them receiving pollution. We're aware of sewage spills or wastewater discharge, and that seems to be the obvious issue. External nutrient loading can be agricultural runoff, it can be stormwater runoff. So those are the more obvious ones to me, and they do need to be managed, both ex situ and in situ. Something that's less spoken about is internal nutrient loading. So what I mean by internal nutrient loading is picture a dam like Hartbeespoort. It has been receiving wastewater and loading for decades. It's been hypertrophic for decades. It's essentially the sort of maturation pond of Gauteng. Huge amounts of organic sediment and therefore a very large nutrient bank. It retains a lot of nutrient, more so than comes in on a sort of daily basis. You have this accumulated portfolio of nutrient sitting in the bottom of that lake. When your water quality destabilizes because of these issues, it starts a negative feedback loop. The nutrients cause the invasive weeds like hyacinths to grow. They cause harmful algae blooms. Those conditions start fluctuating dissolved oxygen levels and redox levels day and night. You get these major swings, and that sediment that is supposed to be stable and sitting at the bottom sort of goes into reflux. Nutrients start coming out. Huge accumulations of this, high concentration, goes back into the water column, and you have a compounded effect. Not only do you have the sort of fresh external nutrient loading coming in, but you've also got this accumulated mass built up over decades, donating its nutrients back into water column, and therefore your algae gets worse, your invasive weeds get worse, your dissolved oxygen levels demand increases, and we have unsightly, dirty, smelly lakes with algae and or weed problems. So our job in recalibrating a water body is to manage both that external and internal nutrient release. We want to switch off internal nutrient release. We want to reduce those numbers, deal with, give the water body the ability to cycle and take care of that fresh pollutant load so that you can have reprieve from harmful algae blooms and invasive weeds. And then that picture starts coming back again of the way the water cycle is supposed to work. So it's about reversing the aging and restoring natural aquatic function. We're not doing anything artificial here. We're not trying to dose it with chemicals. We're not trying to get it to change overnight. It's a recalibration effort to turn back the aging. A water body has got an inherent ability to look after a certain amount of nutrient and carbon. Fish waste and decaying leaf matter. The biology is there in a healthy system. Our surface water systems have lost that ability. So just briefly, key nutrients, the nitrogen and phosphorus challenge, this is the main one. So you have all of this coming in on a constant basis. By keeping that water body oxic with a healthy redox level, we're able to enhance the biological cycling of ammonia nitrogen and support phosphorus uptake through biology, but so to sediment and lock up phosphorus levels. So phosphorus is a persistent thing. What we want to do is get biomass to take it up, and what's not taken up, you want locked up into the sediment. You don't want it coming out again. This is the way a healthy lake works, and this is what nanobubble injection restores in a lake system. So yet again, just talking about back to sort of biology, and if you were to throw a lot of unknown enzymes or bacteria into a system, you might start breaking down organic matter and releasing ammonia and phosphorus. That's then going to trigger a bad cycle of algae blooms and invasive weed growth. If we keep things oxic throughout that water column, top to bottom, through the use of oxygen nanobubbles, we will naturally sequester and reduce those phosphorus levels and cycle the ammonia nitrogen. So we give the biology the helping hand it needs, the tools it needs, sort of the paycheck for the bacteria, and they're able to get to work and do what they're supposed to do, or do what that system is supposed to do, which is take that ammonia and ultimately gas it off as nitrogen. So we're able to do this in virtually any scale water body with shore-mounted equipment, supporting microbes. It's about breaking that cycle, turning back the clock, dealing with the issue from sediment to surface. Sediments are a key contributor to our issues. Putting an aerator at the top or throwing some reagents into the water and sort of bringing all that sediment up can be very, very problematic. We've got to take care of that water body from sediment to surface and really just adapt and restore the function that water body is supposed to have. All right, moving on to a little bit of wastewater quickly. We see obviously a lot of challenged wastewater works. Something that's happened over the years, if you look at a wastewater treatment plant that was designed 40 years ago, they might have designed that plant for 400 or 500 COD, milligrams a liter, and maybe it was designed for 20 megaliters a day. That plant is now receiving, for example, 1,000 COD, and maybe it's not even hydraulically challenged. Maybe it's still running at 20 meg. But our wastewater as humanity has become more difficult to treat. The reason it's become more difficult to treat is our chemical consumption. We look at not only food and manufacturing industry, and the sort of cocktail of chemicals they need to use to disinfect and keep things clean, but so to our own homes. We can think back to the days where we might have had a bar of soap at the sink or the shower. Now everything's liquids. My wife has got 8 different lotions and potions for hair and body, and these are all going down the drain. They've all got surfactants in them, and they upset process. All right. A surfactant's job is to encapsulate and remove soiling. That's the opposite of what you want happening in a wastewater treatment process. A disinfectant that you use, its job is to kill bacteria. That's the opposite of what you want happening in a wastewater treatment process. So the water has become more difficult to treat. The biology is sensitive to this. And the plants are not operating the way they're supposed to. We learned we can use nanobubbles as a sacrificial particle, right? We inject nanobubbles into raw wastewater before the biological treatment process starts, right? A surfactant has a hydrophilic head and a hydrophobic tail. The nanobubble acts as a collector of these compounds. It reaches the destabilization point and ruptures that surfactant. So that surfactant now does not impede the bugs in the biology that are going to do the work for us. The surfactants also reduce aeration efficiency. So if you have an under-aerated or low DO wastewater system you're struggling with, a lot of the time, the culprit is actually the surfactants and fogs that are in the system, impeding the transfer of oxygen to biomass. Yet again, nanobubbles get to work, sequester that issue out for us so that the rest of your process is able to work the way it's supposed to. What that means in industrial and municipal wastewater treatment is better total nitrogen removal, potentially up to 40% less aeration, better solids removal, better separation, even anaerobic digestion. And finally, we're seeing less chlorine use at the tail end of these plants because those surfactants were actually quenching chlorine consumption right at the end, because the surfactant, the majority of them will pass through process. Nanobubbles are getting adopted in irrigation and distribution systems because they manage biofilm and scaling. So these little bubbles, happy to line up on surfaces. They react with biofilm accumulation in pipe so that you don't have that fouling. This helps farmers and irrigation schemes a lot. Similar story with scaling. Soil application, reducing water surface tension with these bubbles, getting better permeability through a soil profile. So Heiner might speak to this when he talks about some of our hydrocarbon work, where we see this in agriculture, we see it in remediation work. We're able to mobilize water, get water to transport through soil better, break up compaction. So you get better water use out of that and better transit through to root zone. Nanobubbles will collect, mobilize, and sort of relocate salts. So if you have compaction or ionic stratification in soil profiles, we're seeing success with nanobubble breaking that up and relocating or immobilizing those sort of salt accumulation issues. All right, on to some case studies. Hartbeespoort Dam. I think some of you might have heard about this project. I will give you the condensed version. Machali's Water was contracted to run a project, multidisciplinary project. One portion of the project was looking at different ways to manage the internal and external nutrient levels of the dam. Obviously, the big visual case for everyone is the hyacinth coverage or the algae blooms. Root cause is obviously the nutrient. So what novel ways are there to deal with nutrient? So enter the Bionomics approach by combining nanobubbles and the correct consortium of microbes. Importantly, this was a pilot on a particular zone of Hartbeespoort. Unfortunately, the public was expecting or hoping that one small pilot was going to fix the whole dam. That was never the case. What it has, however, done, though, is given the utilities and powers that be the confidence in a long-term study to see the change, see the impact. So what we did over that period was achieve measurable reductions in key nutrients like phosphate and ammonia. An immense amount of data was built up. We saw very encouraging changes to redox and dissolved oxygen. Just jumping back, we actually saw major reductions in organic sediments, as much as 74%. So giving us more water column and less nutrient flux. And this is all still biologically linked. At the end of the day, we're sort of detoxifying that water, getting the biology to work again, and arresting the internal nutrient release issues. The findings of this actually exceeded our expectations in terms of the zone. We have seen overall improvements right through to the dam wall, despite the incoming systems actually getting worse. So the Crocodile River has gotten worse, not better, according to Makhali's data. But despite that, we've still seen positive trends in that water body itself. And pleased to say the pilot is continuing. There will be further expansions on this program into Hartbeespoort. We do need to have context for how large and how complex that system is. It is not going to change overnight. This is a program that spans many, many years. It could condense in time with additional sort of inputs and budgets, but the expectation is this will be a program that exists over many years. Rudderplot Dam, this was an incredible project. Kind of got approached by World Rowing. Rudderplot was set to host the first World Rowing Regatta on African soil back in 2023. And there's 2 wastewater treatment plants directly impacting the dam. It's got a long history of eutrophication and problematic algae, and water quality spiraled out of control leading up to the event. So about 3 months before, we were contacted to say, can you pull a small miracle off here? There's 3 months to go. And they were failing on E. coli and various other parameters. And that prestigious event that the city of Tshwane and the country was very excited to hold was under threat. Blueplanet got to work. We sort of threw everything we had at this, ran a really, really intensive program. I think it's managed to give Heiner a few gray hairs as well as everyone else. It was sort of round-the-clock effort for weeks and months on end. And yeah, managed to save the day despite huge activated sludge ingress from a wastewater treatment plant. It was the height of load shedding, sludge spills were occurring, everything was against us. But combining nanobubble technology and the right consortium of bacteria and enzymes, we were able to get things in control and get them in compliance for that event. So yeah, that was sort of the first big public project that gave people some awareness around the technology and the approach, and things have grown since then. So yeah, that was one of these sort of short-term interventions. Unfortunately, that was limited to funding. We contributed, some other consortiums did, to try and get that done. Fortunately, budgets for continued management of the dam as a whole have not yet come about. So it was the sort of treatment effects were limited to event, and to this day is sort of around the jetty zone.
Sorry, Rowan, you asked me to give you a 15-minute heads up. This
All
is
right, thank you. Thank you very much. All right, so another interesting project, lakes and dams are one thing. You have a certain amount of retention. What if you have a flowing river? A really, really toxic soup of sewage and chemistry. This was the case for Greenways Golf Estate in Strand, Western Cape. I arrived to this middle picture. The Soot River has is severely impacted upstream by an informal settlement that particularly sprung up over the COVID period. There is no services there, so the river is used as a sanitation dumping ground and debris dumping. This then carries on down through some underground culverts. It affects a few communities before that, and then pops out at Greenways Golf Estate, which is a coastal golf estate. Picture yourself retiring, going down to the coast, setting up home in a lovely golf estate. You take your grandkids fishing in the river. Life is grand 20 years ago. Over about the past 6 or 7 years, this river is turned into a septic, very sort of dangerous system. Residents were having to close up keyholes, and were putting towels below doors to prevent the noxious odors coming into their homes. The river was completely dead. They'd tried a lot of things. They'd tried aeration, they'd tried chemistry, they'd tried enzymes. Nothing was helping. And the city couldn't get the problem under control in terms of upstream. This picture is an aerial shot, quite a famous one, of that river looking blue-grey. We picked up portaloo dumping, formaldehyde, all sorts of things. This is what their sludge and solids look like. So we got asked, what can you do? We acknowledged how complex this is. We could only work within the bounds of their estate. So this is where the water starts, goes through, and there is housing virtually 50 meters from the start, and odor is everywhere. How do we get a grip on this with limited budget in such a short space of time without going upstream? So we deployed a sort of novel way of integrating the nanobubble technology and injecting microbes and creating some retention. This went to a committee vote to raise a special levy to pay for. After 3 months of proving the point, there was 87% vote for yes, for everyone individually to pay for the effort. And besides just getting rid of odor, we managed to make some serious improvements to the water quality itself. Here's just a visual of what happens within about 250 meters of where our impacted driver is. So very challenging condition, not a lot of time, not a lot of budget, but we managed to make a big difference by sort of using novel bioengineering techniques, the right approach with technology and biology, and managed to make a big difference for the resident and the ocean. So we created we did quite a bit of recirculation. One example is pumping water from the lower end back up to the start. We constructed a biochar activated filter pond. This is where we keep most of our biological activity alive. Another challenge we have is salinity ingress. So the tide comes in and we go from low saline to high saline. We established this pond. We introduced nanobubbles, microbes, and have some retention with biochar over there. And this has got them the abatement that they need and fairly serious reductions to critical water quality parameters as well. This got the attention of the city and other stakeholders and catchment management forums. I'm pleased to say that these programs are now being expanded and rolled out. So what started off as a small niche challenge, the project sort of scaled and grew in terms of outcomes and expectations. And we're pleased to say we're working on other much larger projects now with certain municipalities. Another quick example, I can't name this lake yet. It will become public in the next few months. But I couldn't resist sharing some of the data insights. So it's a 6-hectare hypertrophic lake. We deployed oxygen nanobubbles and our bionomic balance strategy. In a short space of time, 82% reductions in ammonia, 99% reductions in E. coli, and a complete shift in oxygen and redox dynamics. So here is some of our real-time monitoring snapshots. This is over a week. If you look every day, that lake was spiking in dissolved oxygen thanks to photosynthesis and algae. And after hours, it was crashing. Every single night, just before dawn, virtually zero dissolved oxygen. So can you imagine the stress on the species and the biodiversity where you go from high to zero every single day? Halfway through the project, this is what dissolved oxygen looks like. We're keeping virtually everything above the sort of anoxic zone, and this curve is just improving all the time. Similar story with redox. So here, these are negative ORP conditions, sort of undesirable microbial reactions before. And here we are during intervention, keeping everything positive and getting control of those nutrients. At this point, I'd like to ask Heine to talk some about some of his projects here. He knows them better than I do. Heine, are you available now?
Good morning, everyone. Yeah, so let me jump straight into it. So this was a project conducted for Anglo American at the Nail Dam, a part of their biodiversity offset area. The dam has been historically heavily impacted by the Mokopane wastewater treatment plant, depositing some 7.5 megaliter a day of very poorly treated and in a lot of cases untreated wastewater into the dam itself, resulting in a very large fish kill and the dam turning eutrophic, hypereutrophic. Nanobubble technology and a biorem program was implemented both at the wastewater plant maturation ponds and at the Nail Dam. And within a 6 to 7 month period, the dam was back to its natural condition. There's an ongoing program at the dam to ensure that it remains pristine. One of the biggest challenges here was the amount of bottom sludge accumulation within the dam from overloaded maturation ponds, and during every storm ingress into the wastewater treatment plant, the blowouts that were occurring were ending up in the dam. The water results on the right-hand side show you the quality of the dam. This was in 2022, but the results are very similar today. Next one, please, Rowan. And we'll talk a little bit about hydrocarbon spills. We focus on very remote hydrocarbon work, basically where it's impossible to remove soil from a site. This case here that we have in front of us was in Mozambique. It was a 300,000 litre diesel spill of a train derailment, train falling down the side of the hill into a very remote area on the riverbanks, and obviously leaching the diesel into the substrate, into the soil, into the rock bed of the tracks, and then down towards the watermark. Some of the diesel made its way downstream. This needed that booms were to be installed to contain this and bring it to the side, where it was where we used Koya to obviously absorb a lot of this product before bioremediating the area once the floodline had receded, as this event occurred during heavy rainfall. The lab results, I think, speak for themselves over the period of time. This was achieved in 4 months. And here we applied a very, very easy and basic system where it was about containment, then identifying the site and understanding what the topography looks like and what the actual plume into the area looked like. And based on that, the program was developed to deal with the hydrocarbon contamination. A grid drilling program with augers was implemented, and a flush with a hydrocarbon encapsulator was undertaken, encapsulating in a silicate, after which a pH correction was done, and then nanobubble water, oxygen nanobubble water, together with nutrients and a bioremediation program. Once this carbon, nitrogen, and phosphate balance was achieved, was introduced, and we were able to bioremediate this in situ in a period of 4 months. Similar occurrence was in Komatipoort, another train derailment. It was actually an accident. 2 trains collided. This was back in 2023, and exactly the same occurrence happened. That it was a very similar plume, only here there was wetlands along the side of the river, reed beds, and shallow water. So that took a bit of a different approach, but we were also very, very successful in remediating this, again, by applying a number of technologies. Here we can see the damage caused to the environment and the seepage that took place, obviously entering into the river, and then this would seep downstream and affect the Kruger Park per se. Next slide, please, Rowan. Here's some of the surface applications that we did using encapsulation technology, both in the wetlands. This product does have a very high pH, so that needed pH correction again after the program, after which a microbial bioremediation program was applied. We also did build trenches, line trenches, to stop any seepage down to the river and the wetlands areas, and that was then removed from site using absorbents to take up whatever hydrocarbons were left in the process.
Thanks very much, Heine. Yeah, incredible work, especially in the remote areas. And yeah, really refining the acclimatization process and the microbes, how we introduce them, and then using oxygen nanobubble to help that delivery even into substrate. I'm sort of out of time, so I'm just going to take 5 minutes to wrap up now. Challenges and cautions. All right, so challenges we see and sort of we face on a regular basis. Sometimes I get frustrated that we know we have the tools, the technology, and the know-how to fix so many challenges out there. What's obviously lacking is project funding. Public-private partnerships are needed, but it's difficult getting these unlocked. We hopefully have a few interesting ones on the horizon for us. Another challenge is the sort of diversification of the pollution crisis and the catchment regions. So you have multiple municipalities and catchments. Some are impacting others downstream. So, you know, there's debate around whose problems these are. And yeah, that's a day-to-day challenge for us, especially with the river systems and dam systems, where we need to move upstream, deal with sources, start rehabilitation so that you can have the benefits downstream, but it becomes a bit of a bun fight in terms of whose territory, whose budget. Another sort of regular occurrence is what I call FUD, fear, uncertainty, and doubt over biotech and sort of anything considered non-traditional. The kind of work we do isn't yet in the textbooks. So some decision makers who sit with sort of traditional academia or engineering standards, this is novel, it's new. In some cases, it's difficult for larger utilities or entities to sort of tender and procure and get these projects in. There is also a traditional preference for chemicals and sort of commodity OpEx, the status quo of how things have been done for decades. The reality is our problems have compounded, the challenges are different, and the toolbox we have at our disposal is now different too. We can do things we couldn't do decades ago. Public perception is another one. The water cycle and water quality is incredibly difficult to understand. Hartbeespoort in particular, when the public who have suffered a lot with the condition of that dam hear about a new project and funding and millions of rands spent here and there, they've seen a lot of wastage over the years. They want success. They want things turned around fast. It's difficult for them to understand the challenges at play, the water cycle, and the scale of these problems. So managing expectations in terms of turnaround is something that needs more work, especially from the public sector, and to sort of manage that institutional mistrust that's been developed over years. Cautions for us and those in the space. Silver bullet products, snake oil. There's a lot of over-promising and under-delivering. It's challenging for us who might work in similar spaces, saying bugs don't work or oxygen doesn't work. There's a lot of over-claiming out there, so be careful what you're told. Look for evidence, dig into the science of it, understand the complete mechanisms of what's happening with a claim, and realize that it probably takes more than one technology or approach. A lot of these projects, you've seen our Bionomics approach. It's not just one, there's no one silver bullet. It's how we integrate, how we manage and monitor that. There is a sort of unfair expectation that we expect the environment to be our wastewater treatment plant. Attenuation and dilution is one thing. The network's been overwhelmed. We should not be expecting our rivers and dams to be the wastewater treatment process. We need to consider those rehabilitation events, and you still need to get back to the root source of why it got to that. Understand scale. We get asked to look at various projects. Clients sometimes think a 5 hectare and a 1,000 hectare job, the 1,000 hectares is not going to cost much more. The timelines, the scale of the funding is very, very different. Another message I like to put out there is in terms of pollution, water management, restoration, the sort of long decision-making cycles, access to capital. What is the cost of doing nothing? Quite often the answer to that is the costs of sitting back and waiting are compounding, making these rehabilitation efforts even more costly. I'm not going to run through all of this, but just so you know, things we're currently busy with or working on, mass adoption of pre-treatment into the municipal industrial wastewater sector to basically get those plants to do more with less, whilst we wait for those long engineering timelines of upgrades and new works. We've developed and are looking at implementation of our pre-oxidation strategy to save drinking water plants from eutrophic sources, larger and larger lake and dam projects. I've got a keen interest in looking into scaling and optimizing particularly constructed wetland footprints. Constructed wetlands are an excellent form of nutrient removal and water treatment. However, we often underestimate the footprint behind these things. And it's looking like some of these approaches could potentially increase the footprint of constructed wetlands by 75%. We want to see more and more data out there, getting real-time monitoring into public water systems and waterways, and getting communities, public and service providers having access to tangible trending data in virtually real time. All right, that is me. I'm just leaving this one slide just to remind us that nanobubbles are unique. They don't rise out of water. Thank you very, very much, and look forward to the Q&A.
Thank you very much. I must say, I think the amount of people still on the talk, and you filled the talk quite nicely. Thank you very much. Teresa is going to enable the mics. I think start with the Q&A questions if it hasn't been answered, and then some of this on the chats. But then if anybody else has a question, please raise your hand so that Rowan and Heiner knows what type of sequence to answer that. I also just want to say thank you very much. We have covered the whole range of nutrient releases, of bioremediation. We've got a few new terminologies. We have realized that we need to know what we buy and bug-wise, and how to aerate systems. And now we know what a nanobubble is and how special it is. Thank you very much. So if there is any questions, Teresa will enable the mics now. Any raised hands?
I believe that there are some questions still to be answered in the Q&A section, Rowan.
Okay, yeah.
Professor
Right.
Esther van Heerden had a question. I've answered some of the questions. If you'd just like to run through those, please. And then there in the chat, there's also a number of questions that still require your attention.
All right, a question from Conrad on flowing river systems compared to static water bodies. So yeah, speaking to the polluted rivers and how they impact generally as a source of sewage in these things. So yeah, that's why we developed bionomic flow. And you saw some of the work in that Soot River story. Rivers are very, very different, obviously, because of that flowing nature and the hydraulics compared to stagnant water bodies or long retention systems. So the techniques of how we calibrate and how we integrate are different. What we generally look for is any possible, even short-term retention in a system. So attenuation, retention, detention ponds, side stream systems, that gives us the ability to sort of amplify the treatment effects. We also introduced large amounts of surface area. So we integrate high capillary, high surface area biochar into canals, rivers, streams, and detention ponds. So within the space of about within the space of about 200 liters of volume, we can create 20 square kilometers of surface area, and that becomes habitat for beneficial microorganisms and reactions that we want to happen in that water. And generally, the first things we see in a river is a reduction of odor. We start switching off the sulfur-reducing bacteria mode, start the ammonia nitrogen cycle, dealing with the mercaptans and other odorous compounds. Because what tends to happen with these polluted rivers is they're in such a toxic state with so many chemicals and surfactants that biology isn't really functioning. It's struggling to do any natural degradation. So we sort of restore that, detoxify it, switch on the biology again, and the effects start from that treatment point. So it obviously gets better and better downstream in an accelerated fashion. So yeah, long and the short of it is there are approaches to river. There's a lot of consideration and sort of bioengineering works around it, but the strategies are there, the work is there. Nanobubbles in flotation, particular copper flotation, to help boost recoveries. Yes, so there is a lot of work in that space, various kinds of mine process and other separation, flotation, and sedimentation work. The nanobubbles basically act as a bridge. They enhance existing coagulation. They collect fines at the nanoscale that chemistry and normal bubbles don't normally get to. They then group up with a coagulated particle or larger bubble, and then move to flock or flotation. So it's very niche work, but yes, the bubbles are used in that process. It's not biological. We're using them for their physical, mechanical traits. Another question is, will the technology work where specific microbes that thrive in extreme low pH environments, such as acid mine drainage? So there's quite a few niches to unpack in that. If the processes are meant to be anaerobic, in other words, if you're encouraging anaerobic or facultative, we can keep it in that direction. We can use a different selection of gases or lower the mass transfer rates and use the nanobubbles to stimulate the biology. So it's almost like a sort of steroid to the biology. If it's a beneficial microorganism, we can increase its functionality potentially. We obviously can't speak to every single microbe out there, but you've mentioned a few there I would like to look into and explore further. But we do this in anaerobic digestion as well. So we remove some of the toxicity, we enhance the enzyme production and enzyme kinetics and microbial community health. Also in acid mine drainage, we can use nanobubble technology just for its oxidation efficiency and potential. So we can use ozone nanobubbles to optimize oxidation and therefore separation of metal and particulate as well. Another question on, has it been trialed with recovery systems such as wetlands? Yes, there is wetland work in South Africa, more along the rehabilitation of wetlands that have sort of been overwhelmed with solids and muck and microbial function is decayed. So we see a waking up, stripping of problematic loading in those wetlands and a restorative function. What we're still wanting to get more into is the dynamics of specifically how much better we can get a wetland to function in terms of its removal rate. Yeah, very keen to do more work on that. We believe there's a lot to unpack there. I'm just looking for other questions here. How safe are these products on receiving environments and organisms, especially when used in nature reserves and or protected areas? Good question. This is something we've been asked by environmental specialists and departments and municipalities. When diving into this, the sort of long and the short of it is, we only provide a consortium of bacteria that does exist naturally in soil and water in this country and around the world. All right. The difference is we have them available in numbers. We have them available to produce enzymes where necessary, and we have them virtually on tap, and we can introduce them into environments that are maybe stressed or don't support the right conditions yet. By introducing that, you're not doing anything unnatural to that system. Those organisms could very well exist there. If they are not needed or they don't have work to do in that environment, or that environment can't support them, they simply die off. So yes, we have, because we can identify and because we know exactly what those organisms are, a institute could look at it, validate that they're comfortable with that speciation, acknowledge that they are sequenced, genome libraried, and very well studied. So yes, we don't have challenges with that, and very, very comfortable with the results and work over many decades. All right, I think I got through the questions in the chat. Open to any more that come through.
Rowan, there are some more in the Q&A section, particularly the statement from Professor Esther van Heerden. If you could just unpack that, please. That is the third from the top.
Okay. Is there any other questions? If there's not,
Sorry, I was busy talking to myself there. I had muted.
Sorry, yeah,
was
it
just
was so quiet. I
through.
was just
Sorry.
wondering what's going on.
A question from Esther van Heerden. Introducing foreign bacteria under the Biodiversity Act, how do we ensure that it does not impact native groups? I feel strongly that we should stay indigenous and do extensive permitting for site-specific bacterial groups that must be custom for specialized contaminants like PCE. Yes, this is a very valid query. This is why we have tools like Nanobubble technology at our disposal. If there are concerns in a key application or highly sensitive environment that cannot be appeased with a certain sort of packaged or external microbial consortium, if comfort factor is not reached there and you don't want to introduce what may be considered foreign, then we turn our attention to encouraging natural microbial habitat and sort of strengthening the microbial community. So if possible, we would look at introducing nanobubbles into that environment to appease anoxia or perhaps negative redox conditions or anaerobic conditions, if that is the target of the particular application, and really wait for the natural microbiome or the natural microbial communities to shift to what they need for that environment, which is essentially what we sort of do even if we apply. The microbes are going to be competitive to what's needed, the kind of work that is needed there. If they're not needed, as the nutrient or the conditions not right, they're naturally going to fall away or be outcompeted anyway. But we see Nanobubble as a tool to encourage that stimulation and support of what the naturally existing community, or at least the shift, could be. Another question, was the cost for replacing a nanobubble generator to a standard municipality wastewater works done? And will it be worth it to upgrade the current municipality works with this technology? So mass adoption of municipal pre-treatment is in its infancy in this country. We've done private and industrial work. Hopefully you'll hear about some municipal plants going ahead quite soon. It is a very easy retrofit. We do not need to work with an upgrade of a plant, any civil or process changes. A lot of these installations actually take place in about 2 days. The technology is implemented at the head of works, and the plant can continue to operate under standard operating conditions, now with improved oxygenation, improved microbial community health. It's a very easy retrofit. Cost-wise, it generally works out to it obviously depends on the size of the plant and the problems, but we're looking at about 3 to 6 months of their average electricity cost to purchase and implement these systems. So it's really a fraction of their operational requirements. All right. Are there any, okay, another one. Are there any processes occurring naturally that stimulate the nanobubble process? I like that question. I haven't been asked that before. It depends, yeah. Thinking about that now, obviously oxygen availability is one that's the sort of more basic metric that will dictate what your process looks like in a natural system. You have influences over weather, temperature, wind, plant cover, stratification. Those are the major driving forces between what oxygen levels look like and how the biology performs in a system. The nanobubble, on the other hand, the actual functional nanobubble itself, they can occur naturally. That's how they were discovered. Ocean crashing waves, waterfalls. We see this fractionation of nanobubbles there, and that enhances process. Do nanobubbles occur naturally in something like a lake or dam? No, we don't believe so, because there's no sort of shear or mechanical forces to induce that. All we know is the biology and the natural world work with them, they like them, and we're not yet sure of another way to sort of stimulate that same process because it's very much multi-mode. It's the gas provision, it's the behavior, it's the mechanical traits, and the biogeochemical reactions. Another question, apart from wetland, how do you remediate hydrocarbon contaminated groundwater? Have you done any work on this? And what was the outcome? My experience is limited with sort of in situ groundwater, but the similar principles would apply. If abstraction is possible, obviously that's a lot more straightforward. It's about fine-tuning the nutrient profiles, microbial community, and the support systems to the biology that would degrade the hydrocarbons. Sort of down well or within water table, we have heard of, and there are, I believe, studies on injection, on microbial introduction, a little bit more complex. So I don't have exact experience with that, but I believe some international work has been done. Okay, I think I've got all the questions for now.
Yes,
let me know if there's anything else.
I think that is it from us now, but I am sure there's a lot of more questions from people. Your contact details are in the chat, and then also the registration for the Coltech colloquium. Even if you want to showcase your work, the consultants in our audience, there's registration links for both participation and exhibits. So I think that's it from us from LaRSSA, but Rowan and Heiner, thank you very much for sharing this crucial information. It will help us evaluate certain technologies in future, and we hope to hear from you again. Thank you very much.
Thank you.
Thank you all. Thanks for the opportunity. Yeah, please do reach out on mail to Heiner or myself. Thanks all.
Thank you. Bye.